Yeah. Okay, welcome back to the last talk of this session. It's my great pleasure now to introduce to you Dr. Roshan Cools as our next speaker. Dr. Cools completed her undergraduate degree in experimental psychology at the University of Groningen in 1998. She then moved to Trevor Robbins' lab at the University of Cambridge, UK, for a PhD degree, followed by a Royal Society Research Fellowships until 2007. She also spent 2 visiting postdoc years at UC Berkeley, working with Mark Desposito from 2003. In November 2007, she returned to the Netherlands, where she is now principal investigator in the Dondes Institute for Brain, Cognition and Behavior and full professor of cognitive neuropsychiatry at the Radboud University Medical Center in Nijmegen, studying the chemical neuromodulation of human cognition. Dr. Cools has made lasting contributions to cognitive neuroscience, providing empirical foundations of current theories of dopamine and serotonin's role in human motivational and cognitive control. She has pioneered the combination of advanced techniques, including pharmacological fMRI, chemical PET, psychopharmacology, and computational cognitive science. She is an elected member of the Royal Netherlands Academy of Arts and Sciences and the Academy of European. Yes, it's my great pleasure to have you here today, and we're looking forward to your talk. Please. We can't hear you, unfortunately.
Yeah, sorry.
Okay. No, it's
always
good.
The same problem.
Welcome.
Yeah, I'm really excited to be here. Really enjoyed the previous talks by Evelina and Angela. Let's see, can I share the screen? So can you see my full screen?
Yes.
We can.
Okay, great. Yeah, and just to follow up, it's always sad that we can't actually interact, but I did want to just follow up that on Angela's talk, that this new technique of ultrasound neuromodulation, focused ultrasound neuromodulation, is something that we're pioneering here, and it really provides this great opportunity to start intervening with these deep regions in the brain. So I'm quite keen to do some of that and maybe use some of the similar paradigms. Anyway, I'll talk about something different, dopamine. And I just want to get myself out of the way. Yes, so I can see my own slide. I just want to begin first by acknowledging my wonderful team and the fact that all the studies I present are the result of collaborative effort, of course. I'll focus in the first half of my talk on work that was done with Rut van Holst, who was a postdoc in the lab, and Hanneke ten Ouden, who's now a PI here, colleague at the Donders. Also Mirjam Bloemendaal was a PhD student in the lab and now postdoc in a colleague lab, and Martine van Schouwenburg. And then in the second half of my talk, I'll move on to presenting work from Andrew Westbrook, who's currently at Brown University. It was work in collaboration with Michael Frank at Brown, and work by Lieke Hofmans, who was a PhD student, currently postdoc in Amsterdam. And all of that was done really with assistance in a larger team, including other people that I highlighted here in pink. Okay, so I think it's fair to say we live in the age of the connectome, where the brain is conceptualized as a distributed computing system comprising multiple densely interconnected neural networks that can be characterized with exquisite detail. But a key point is that these connections, these structural connections, are relatively fixed, at least in the adult brain. So a major question is which mechanisms allow our brain to adapt so flexibly to its constantly changing environment. And the major ascending chemical neuromodulators that originate from the midbrain, like dopamine, turn out to be particularly well suited for this. So it's the chemical neuromodulators that adapt the output of our structurally fixed networks to our changing environment. Indeed, the brain constantly faces a variety of computational dilemmas, depending on task demands. These networks, these neural networks need to exhibit stability or flexibility, controlled processing or automatic processing, speed or accuracy. And these dilemmas are at the core of cognitive control, which is an ill-defined construct, but generally refers to those mental processes that allow us to obtain our goals by resisting distractions and temptations, impulses. It's a hallmark of the human brain, of course. And so I guess you won't have any problem to continue to listen to this talk, third talk in a session for the next 50 minutes, right? Of course, the question is to what degree you will decide to make use of that wonderful ability to focus and exert control. Because after all, control is costly, intrinsically costly, but also opportunity costly. So some apparently distracting messages actually turn out to be quite important and require that you let go of focus and control. We'll see. What we need is a meta-level ability to decide when to exert focus and when rather and control, and when rather to let go of that focus and let go of that effortful control at the right moment. So how do we do that? And answering that question requires that we reconceptualize the problem of cognitive control as a problem of motivation and decision-making, instead of simply as a problem of implementation, of top-down implementation. And this take-home message that the chemical neuromodulators, including dopamine, adapt the output of our networks to our changing environment is grounded in part in the observation that they implement this decision-making ability, this motivation ability. So the arbitration between distinct, often opponent computational strategies. Okay, I'll begin by highlighting, well, I should probably say that I'll focus on dopamine, of course, and then begin by highlighting an important problem in neurology and psychiatry, namely that there's really huge variability in the effects of dopamine and dopamine drugs, both across different and within different the same individuals. Next, I'll illustrate how we have begun to account for these paradoxical effects, for this large variability in the past. So I'll take you with me on a quick tour through previous pharmacological and chemical imaging studies that really represent two lines of work, one focusing on striatal function, so motivation and decision-making, and the other mostly on frontal function, cognitive control and working memory. And then the second part, I'll show you some more recent work where we begin to integrate these dual roles of dopamine. And then I'll end by raising a number of open questions that arise from this work. Okay. Cognitive role of dopamine is, of course, keenly illustrated by its implication in a wide variety of neuropsychiatric disorders, ADHD, schizophrenia, Parkinson's disease, various addictions, but also healthy states of stress and fatigue. All these states are accompanied by cognitive and impulse control deficits. And critically, these states can be treated or are often treated with drugs that alter dopamine transmission, also in healthy states. Unfortunately, there's huge variability in the effects of these drugs, as I just said. So, for example, consider ADHD, where symptoms of inattention and impulsivity are often treated with dopamine-enhancing drugs like Ritalin, methylphenidate. And conversely, consider Parkinson's disease, where dopamine-enhancing drugs instead can contribute to cognitive control deficits, leading in some cases to severe psychiatric abnormalities, including addiction. Various of the behavioral addictions are seen: gambling, punding, compulsive behaviors. So one of the key questions that drives much of our work is how can it be that the same neuromodulator has such different effects in different individuals? So let me illustrate how we've begun to address that in the past. Until recently, the dopamine community was relatively split, with some research lines focusing on striatal functions, reward, motivation, learning, choice, and other lines, separate lines, focusing on prefrontal functions, working memory, cognitive control. Of course, beginning with the seminal work by Patricia Goldberg-Keach. Now, striatal dopamine, perhaps best known today for its contribution to reward prediction error-based learning. And in this context, we've contributed some foundations to the hypothesis that dopamine contributes to reward-related compulsive choice. And that line of work, early work, started with a key observation that dopaminergic medication in patients with Parkinson's disease can induce an impairment, paradoxically, on probabilistic reversal learning, an experimental model of compulsivity, if you will. I'm not going into the details of this very old work now, apart from just saying that. And that work subsequently was followed by chemical PET imaging study work demonstrating that the degree to which a dopamine drug, in this case the catecholamine reuptake blocker methylphenidate, Ritalin, impaired performance on this same probabilistic reversal learning task, and that impairment depended on the degree to which that drug increased striatal dopamine release as measured with raclopride PET. So the more methylphenidate decreased the raclopride binding potential, so increased dopamine release, the greater the reversal learning impairment was. So that was in line with this detrimental effect of dopamine medication contributing to compulsive choice. So next, somewhat more recently, we established in a moderately large online study a potential computational mechanism underlying dopamine-related reversal impairment, underlying dopamine-related compulsivity. And in this study, which was analyzed by Annika and involved collaboration with Nathaniel Do also, approximately 700 healthy volunteers for whom we had available saliva samples for dopamine genetic analysis completed again this same probabilistic reversal learning task at home. And what this study revealed was that carriers of the 9 repeat allele of the common dopamine transporter polymorphism exhibited increased perseveration on this task. And critically, we were able to account for that perseveration using a reinforcement learning model that was augmented with an experience weight parameter, which essentially reflects progressively increased reliance on previous reinforcement, capturing the stamping in of a habit with repeated experience. So in essence, the increase in experience weight here corresponds to a progressive decrease in the learning rate with each reinforcement, and so decreased impacts of prediction errors. Now, the key role of dopamine in compulsivity was further substantiated by the observation that striatal dopamine synthesis capacity, measured with FDOPA PET imaging, is increased in patients with pathological gambling or gambling addiction, as we say now, compared with healthy age and education matched controls. And intriguingly, these same gamblers, and now this is unpublished work still, also interestingly exhibited increased perseveration on the same probabilistic reversal learning task. An effect that here was captured by an increase in this same computational parameter, in this experience weight parameter in the gamblers relative to the controls. So here I'm not plotting that effect, I'm just plotting the increase in errors as seen after a reversal. So increased in perseveration errors in the gamblers versus controls. Now, together, I realize that's an extremely fast summary, but the take home is really that the work strengthens the hypothesis that at least some of dopamine's detrimental effects on behavior reflect potentiation of reward-driven action selection. Of course, there's also a large body of evidence demonstrating a key role for striatal dopamine in invigorating behavior. So in modulating not so much action selection, but the speed, in modulating the speed or vigor of responses, in activating behaviors. In particular, in one influential line of work, Yael, Niv, and colleagues have integrated work on action selection and action vigor in a formal model that incorporates a signal for an agent's average reward rate. And in that model, if the average reward rate, so the average reward that has been accrued over time across all kinds of states, currently relevant and irrelevant states, is high, if that average reward rate is high, it's more opportunity costly to act slowly. And that is so because sloth, slow responding, would be associated with more missed opportunities. And that average reward rate in that model would offset the intrinsic cost of acting fast. Now, what's critical is that in that model, the average reward rate is reported by striatal dopamine tone. That was substantiated by a pharmacological study showing that administration of levodopa, that's the drug that's also given to patients with Parkinson's disease, now given to healthy volunteers, boosts the impact of average reward rate on vigor, on reaction times. We've recently built on that prior work and tested another prediction from that model, that individuals with greater striatal dopamine synthesis capacity, so individuals who in a stable manner produce more dopamine, should exhibit greater response vigor as a function of average reward rate, should exhibit greater average reward rate effects on vigor. And so to test that, we had 44 healthy volunteers, just young, healthy subjects, complete an odd one out choice task with fluctuating reward rates. This is actually the same task that was used originally to demonstrate effects of levodopa on vigor. It was a task borrowed from Ulrike Beierholm and Peter Dayan. And so in a separate session of this study, participants underwent an F-DOPA PET imaging scan to quantify dopamine synthesis capacity. And as I said, this provides a relatively stable measure of how much dopamine is synthesized. So uptake of this radio tracer F-DOPA indexes the degree to which dopamine is synthesized in the terminals of midbrain dopamine neurons, in the striatum, right, the striatal terminals. And it's relatively stable across subjects and across, across sessions within subjects. Now, after exclusion of data from a set of extremely fast responders, who basically didn't leave any room for modulation of reaction time whatsoever, we actually did uncover the predicted link, although it was somewhat weaker than we had anticipated, with greater average reward rate effects on speeding in subjects with higher striatal dopamine tone. And what was interesting in this study is that we could localize the effect also to the nucleus accumbens. So whereas previous studies had shown that levodopa indeed potentiates vigor as a function of average reward rate and not instantaneous reward incentive, I should have added, we could here see that that effect of dopamine is localized in the ventral striatum, in the nucleus accumbens. So together, these data converge to suggest that striatal dopamine promotes both the direction and invigoration of behavior as a function of cumulative reward accumulated across history. So to switch gears in the domain of prefrontal dopamine, researchers have focused naturally on more cognitive constructs. course, seminal work from Brzezowski and Patricia Goldmer-Keats had already shown that 6-hydroxydopamine lesions of the prefrontal cortex impaired performance on a classic working memory task almost to the same extent as did complete ablation of the prefrontal cortex. And subsequent evidence from neurophysiological and pharmacological work with animals and also computational modeling work suggested that many of these effects reflect modulation of the stabilization and also distractor-resistance of current working memory representations. So in line with that literature, we established that increasing dopamine D2 receptor stimulation by administration of a low dose of bromocriptine reduces distractor resistance during a delayed response task, possibly via a presynaptic mechanism of action, leading to a net reduction in dopamine levels. And what was important was that this increase in distractibility was also associated with distractor-related disruptions of delay period activity in the dorsolateral prefrontal cortex, shown here in the middle panel on the bottom. So the more that bromocriptine disrupted distractor resistance, the greater were these distractor-related disruptions of delay period activity in a region of the brain that had also been established to be important in non-human primates. And furthermore, the drug-related increase in distractibility was also associated with distractor-related decreases in functional connectivity of delay period activity in the prefrontal cortex, with that in stimulus-specific cortex, in this case, the fusiform face area, because subjects had to memorize the faces. So what we showed here was that bromocriptine disrupts a top-down fronto-posterior mechanism that protects working memory representations against distraction. As such, that study illustrates classic thinking about dopamine's role in prefrontal cortex as modulating the implementation of top-down control of task-relevant processing in more posterior sensory cortex. Dopamine in the prefrontal cortex modulates top-down biasing of task-relevant processing in the posterior cortex. Now, more generally, what this sample of studies, and I realized this went extremely fast, but what I wanted to do was to illustrate that lines of work on striatal and frontal dopamine have really been rather segregated. The obvious next question is, how can these dual roles be integrated? Can these dual roles be integrated? Now, in part, such integration was established in striatal gating models, where dopamine gates cortical actions by increasing activity in a go pathway of the basal ganglia, but decreasing activity of a no-go indirect pathway of the basal ganglia. That kind of computation had been well established in the context of motor control, but that role of striatal dopamine in gating had also already been proposed to extend to the flexible gating of cognitive actions or cognitive tasks or representations maintained in working memory by Hazy and Frank and O'Reilly. So in essence, these cognitive gating models represent an integration of classic top-down biasing models of cognitive control with classic models of the basal ganglia, according to which striatal dopamine lowers the threshold for gating actions. And so inspired by that theoretical work, we set up a series of studies to test the hypothesis that the biasing by the prefrontal cortex of posterior cortex is controlled by the striatum, so that task-relevant posterior processing is flexibly updated only when striatal dopamine opens the gate for attentional switching. And so that's work done with Martine van Schouwenburg and also again Annika ten Ouden. And in these experiments, what Martine did was present subjects with bidimensional stimuli consisting of an overlapping face and scene, And on each trial, in fact, subjects had to discriminate between two of these bidimensional stimuli based on either attending to the faces or attending to the scenes. And they were instructed here to switch attention between faces and scenes as soon as they detected a change of exemplars in the irrelevant stimulus dimension. And what we found was that these switch trials, so as soon as these novel exemplars were introduced in the irrelevant, currently unattended dimension, or currently irrelevant dimension, we found highly significant bold signal, indeed in the basal ganglia, also the thalamus and the frontal cortex. And moreover, as would be predicted, not so surprising, there were also strong attentional gain effects in stimulus-specific posterior cortex, sensory cortex. So with the greatest signal in the fusiform face area when people switch to faces, and greatest signal in the parahippocampal place area when people switched attention to scenes. Now, the overall pattern of bold signal was nicely captured by a dynamic causal model in which striatal switch related activity modulated effective connections from the prefrontal cortex, inferior frontal cortex, to task-relevant visual association cortex. And these data were consistent with the hypothesis that the striatum guides attention switching by gating the cognitive output of the prefrontal cortex. But of course, we're interested in dopamine. And that's why we followed this up with a separate drug study, which revealed that administration of, again, bromocriptine, this dopamine D2 receptor agonist. Again, it's also a drug used in Parkinson's disease, but in this case, we use a small dose. This drug increased selectively switch-related bold signal in the basal ganglia. And what was interesting was that that region was located, localized immediately adjacent to a region that exhibited a link between the behavioral index of attentional switching, that's here plotted on the y-axis, and white matter integrity indexed by fractional anisotropy measured with DTI, diffusion tensor imaging. And moreover, probabilistic diffusion tractography from that region in the basal ganglia showed that white matter fibers ran from this white matter region around the basal ganglia to the prefrontal cortex. In fact, this white matter tract that originated in that basal ganglia region, white matter region, culminated in a region of frontal cortex, here shown in blue greenish. It's quite small. I don't know if you can see my mouse actually, but that blue-green region, which exhibited dopamine drug effects on functional connectivity with that striatal bold region that was modulated by bromocriptine. So just to recap this complex slide, what these studies showed was that bromocriptine modulates switch-related bold signal in a region of the striatum that connects structurally and communicates functionally with the prefrontal cortex. So that leads us then to conclude that striatal dopamine potentiates cognitive flexibility by gating attention to a cognitive task rule via modulating prefrontal control of task-relevant processing in posterior cortex, via output gating, regulating output gating of the prefrontal cortex. However, the key question remained, and that was, how then does striatal dopamine set the threshold for gating a cognitive task or rule? And according to these gating models that I mentioned, as well as currently popular expected value models of control, we allocate resources to a cognitive task based on its expected value. So that's based on the benefits and the costs associated with performing that task. And so specifically, we've been working with the hypothesis that striatal dopamine affects cognitive control indirectly by modulating the value of cognitive work. by modulating the motivation to do this cognitive work. According to the opponent-actor learning or OPAL model by Anne Collins and Michael Frank, which is a modern gating model, if you will, striatal dopamine increases the weight on the benefits, decreases the weight on the costs of a cognitive task, again by altering a balance between activity in the go pathway and a no-go pathway, a direct pathway and the indirect pathway of the basal ganglia. Now, the general implication of this hypothesis is that the cognitive effects of dopamine-related disorders and also their treatment reflect in part modulation of the willingness rather than the ability to exert cognitive control. So to test that hypothesis, with Andrew Westbrook and Michael Frank, we employed a cognitive effort discounting paradigm that Andrew developed when he was a PhD student in Todd Braver's lab. And this task allowed us to assess the monetary value that people assign to repeating a cognitive task, a difficult and effortful cognitive task. The experiment consisted of 3 phases. In the first phase, subjects were exposed to a set of easy, difficult N-back tasks, so a 1-back, a 2-back, a 3-back, and a 4-back working memory task. They had to perform these tasks. And then in the second phase, a separate second phase of the task, they had to, on each trial, indicate by button press whether they preferred to repeat after the testing session, so not immediately, but later, the easy or the difficult one of the 2 tasks in return for more or less monetary payoff. So this is just sort of a schematic of that kind of trial. Now, what's important here is that the monetary payoffs, the amount of monetary payoff for the easy task was titrated depending on choice, so that ultimately we could derive an indifference point corresponding to the subjective value that people assign to repeating a difficult task. Now, we also obtained F-DOPA PET imaging data from the subjects who completed this effort discounting task to quantify striatal dopamine synthesis capacity. So those are, again, relatively stable levels of synthesis capacity. And the data from a placebo session in which subjects performed a task after intake of a placebo pill are shown here. Subjective value that was calculated in the critical discounting second discounting phase of the task is shown here on the y-axis. And what you can see is that the higher the score, the higher the value. So the more motivated people were to repeat the difficult task. And as predicted, this subjective value of cognitive control was indeed significantly higher in subjects with higher baseline levels of striatal dopamine. And that effect was centered on the dorsal striatum, the caudate nucleus in particular. Now, this subjective value of cognitive task performance was also boosted by dopamine drugs. So in this study, subjects were not only tested after placebo, but also after intake of methylphenidate, this catecholamine reuptake blocker, transporter blocker, which increases dopamine and also noradrenaline. So low oral dose here, or 20 milligrams. And they were also tested after intake of a low dose of sulpiride, which is a D2 receptor antagonist, but it's given in such a low dose that we think it acts presynaptically to increase levels of dopamine. And what was important here is that these effects depended on baseline levels of striatal dopamine synthesis capacity, so that both drugs boosted value in low dopamine subjects, but actually, if anything, though not significantly, attenuated these effects in high dopamine subjects, putatively in line with this well-established inverted U-shaped relationship between dopamine and performance. Based on the computational OPAL model that I mentioned earlier by Michael Frank and Anne Collins, we could actually make more specific predictions about the effects of dopamine. namely that high dopamine states should be associated with an increased weight on the benefits for choice, so an increased weight on how much money you could win for doing this task, or not win but gain, or and a decreased weight on the cost, so a decreased weight on how difficult it was to do this, a decreased weight on the effort level. So in the third phase of this experiment, Andrew sampled choices on either end of each individual's point of indifference to test that prediction. And by sampling choices on either end of the indifference point, individually defined indifference point, he could plot the full psychometric curve, which quantifies the degree to which high effort selection varies with the difference between the subjective value of the easy task and the subjective value of the difficult task. And Opel model simulations demonstrate that an increase in benefit sensitivity and a decrease in cost sensitivity, this was to simulate the putative effect of dopamine, was associated with an interesting effect on the psychometric curve. So by increasing the benefit and decreasing the cost, this this curve was kind of asymmetrically affected. So dialing on the benefit and cost sensitivity steepened the right side of the psychometric curve, so that greater sensitivity to benefits steepened the curve on the right side, but it flattened the left side of the curve, respectively. So these are simulated data. Now here on the bottom two panels, you can see the actual data. On the left as a function of dopamine synthesis capacity, so that's an individual differences effect from the placebo. Actually, this is all drug sessions together. And on the right, the effects of the drug. So what you can see is that dopamine had exactly this asymmetric effect that was predicted by these simulations, by this model. With high dopamine states associated with greater weight on the benefits, but also reduced weights on the costs. Now that qualitative effect, or that effect that I've shown you on that previous slide, was confirmed, of course, by statistical regression, hierarchical regression analyses, but also interestingly by dynamic gaze patterns during choice, because that third phase, Andrew did something clever. He actually presented the benefit and cost attributes of the choice separately on the screen. And that way he could disentangle whether people were looking at the benefits or rather at the costs of the choice options when making their choices. So he could separate the contributions to choice of gaze at the benefits and gaze at the costs. And that's plotted here. So gaze is plotted here on the y-axis, proportion of gaze from choice onset. Gaze at the benefits in green and gaze at the costs in blue. And it's plotted separately for high dopamine subjects on the top and for low dopamine subjects on the bottom. What you can see here is that for the trials on which the difficult task was chosen, subjects gazed more at the benefits than at the costs than for the easy task. And critically, that effect was greater for the high dopamine subjects than for the low dopamine subjects. This was also seen as a function of drug administration, but I'm not showing it here. So what this work establishes is that the cognitive enhancing effects of methylphenidate indeed reflect, at least in part, increases in striatal dopamine and associated gating of a cognitive task in proportion to its benefits versus costs. And these findings illustrate that dopamine might indeed help us to adapt our environment by arbitrating between different cognitive tasks by altering the weight on their costs versus benefits. Almost done. The findings, of course, raise an obvious next question, which we are addressing in ongoing work. And that question is that if dopamine release regulates the arbitration between different tasks and maybe even different strategies, then of course what controls dopamine release? In the case of dopamine, we can also ask, how do we set the value of cognitive work? This is not a trivial question, given that valuation is context dependent. So for example, a cognitive work is not so valuable when the opportunity cost is high, for example, due to high average reward rate or high value of exploratory actions. And it's also not so valuable if the environment is uncontrollable, just to refer to the previous discussion we had. Now, interestingly, in the pharmacological PET study that I just described, Andrew used a task that actually controlled for opportunity cost or alternative rewarding activities. In this experiment, subjects always had to choose, after all, between a hard task and an easy task. And in this case, the link between dopamine synthesis capacity and willingness to work was positive. This is what I've just said in the last few slides. Now, what was striking was that a rather different pattern of dopamine effects was observed in a complementary cognitive effort experiment that we conducted in the context of the very same pharmacological PET study. In contrast to the Westbrook study, this Hoffman's experiment did not control for opportunity cost. In fact, participants in this experiment always chose between a difficult task. Sorry, I just messed up my animations here, but they always chose between a difficult task or alternatively, a period of rest during which they were allowed to use their laptop or their mobile phones or whatever they wanted, as long as they stayed in the testing booth. So in contrast to the Westbrook study, this experiment revealed, if anything, a negative link between dopamine synthesis capacity and willingness to work. Moreover, whereas the Westbrook effect was strongest in the dorsomedial striatum, the caudate nucleus, the Hoffman's effect was greatest in the nucleus accumbens, the ventral striatum. Now, we might speculate that this reflects a role for ventral striatal dopamine tone in carrying the value of rest. What is clear, though, is that the direction of dopamine's effects as a function of individual differences, but in fact also as a function of drug administration on cognitive motivation, depend on key characteristics of the environment or of the task, including the value, in this case, of alternative opportunities. Okay, I want to end by speculating wildly about one intriguing possibility, namely that these key statistical meta-decision parameters, like average reward rate that I mentioned early on in my talk, but also its controllability or opportunity cost, are computed cortically, say in medial frontal cortex, maybe area 24, which then prioritizes distinct striatal projections, dopamine projections, by setting their baseline tone in an environment-specific manner. More generally, these data illustrate how chemical neuromodulators, in this case dopamine, can adapt the output of our neural systems to our changing environment. And with that, I've spoken for 40 minutes, and I think that's enough. Thank you so much.
Yeah, thank you so much for your talk. We have now some time for questions. The first one is asking whether you could tell how to increase someone's own motivation or own productivity without using any dopaminergic drugs.
That's an interesting question, and clearly our data do not directly speak to this. And this now refers to my ongoing work that I did not present, and also the speculation at the very end is that I think it will be key to estimate these meta-decision parameters, such as opportunity costs, or controllability, to know when to exert effort and rather when to let go of effort. Because I don't think motivation or doing one's best is appropriate at all times. And we'll have to do more experiments to assess that. And there might be definitely cognitive ways of going about that, rather than simply, taking pills. I'm definitely not suggesting taking pills to boost one's motivation. This work is really to demonstrate the mechanisms, the brain mechanisms underlying such modulation. Yeah.
Thank you. We have another question from Professor Roberts. You've shown how striatal dopamine contributes to prefrontal control through gating, but how do the two dopamine systems themselves interact? Previous old studies suggested there was a seesaw relationship with low prefrontal dopamine resulting in increased striatal dopamine, for example.
Yeah. And I've thought about this in the past also. I didn't talk so much about that, but indeed, Angela's referring to work also with marmosets, and actually similar seesaw effects or reciprocity is seen in patients with Parkinson's disease, for example, where there's not only, particularly in the very early phases, I should emphasize, there's not only striatal dopamine depletion in Parkinson's disease early on, but also some people have reported upregulation of frontal dopamine levels. And so there is some evidence, old evidence already, that these might interact reciprocally by increasing dopamine in the striatum, or rather decreasing it, we get the opposite effect in the frontal cortex. And it's a very interesting observation that this role of dopamine in the prefrontal cortex, which I've characterized as increasing stabilization or distractor-resistance of representation, might represent a sort of functional opponent to the role of striatal dopamine in increasing rather distractor vulnerability, or maybe increasing the flexibility of those same working memory representations. And indeed, we've seen in a few studies, I didn't mention that here, that the same drug might have rather different consequences for stabilization or for flexing of working memory, depending on whether it acts in the frontal cortex or in the striatum. Now, of course, most of our drugs, that's a challenge, most of our drugs for human research are rather non-specific. And so inference about their locus of action relies on disproportionate depletion, for example, in patients with Parkinson's disease versus ADHD or other types of disorders, which might rather be characterized by disruption of cortical catecholamine levels. So one hypothesis is that these drugs might have rather different effects in, say, patients with PD or rather ADHD, because their locus of action is different, promoting on the one hand or normalizing on the one hand striatal dopamine levels in patients with Parkinson's disease, thus promoting or boosting the flexibility of representations, and on the other hand promoting distractor-resistance in ADHD by acting primarily on cortical systems. Yeah, I hope that it goes some way to answering the question.
Thank you. Thank you very much.
Next question is, how does the low-high dopamine in striatum work in case of drug addicts who usually choose high reward despite the high costs, although they should have quite low dopamine in the ventral striatum, at least during the craving phase?
Yeah, so that's a big sort of area of research, right? Dopamine and addiction, and there are competing theories out there about the degree to which dopamine is indeed deficient what's probably important to also highlight is that there's not just sort of one, likely not one mode of dopamine transmission. So while, so I think it's still somewhat unclear to which degree, which mode of dopamine transmission is indeed deficient in which phase of, and which area, brain area, which phase of drug addiction and in which brain area. So while dopamine may be, tonic dopamine levels, for example, may be deficient, there have been some accounts suggesting that dopamine release is potentiated, but there's also the other, the opposite direction. So I'm a little bit hesitant to address directly to let's say, how can I phrase this, to rely on suggestions that dopamine is deficient in ventral striatum and then kind of generalize from that to cognitive effort. I think we have to do these studies basically and assess whether cognitive effort choice is altered in drug addiction, and we also have to take into account what phase of drug addiction and what type of drug addiction. I think this will depend also very much on the target, that on the nature of the benefit that the drug addict will be working for. So some studies suggest that the anticipation of rewards that are relevant to the addiction, for example, is boosted, whereas the anticipation of rewards that are irrelevant to drug addiction are attenuated. So that's another factor that may play a role here and that have to be taken into account.
Are you planning any studies with maybe some patients that suffer from addiction?
We're not running studies with addicts at the moment. We've done this pathological gambling or these gambling addiction studies in the past. So those studies have focused mostly on risky choice. Be quite interesting to look at effort-based choice. And I think a key factor to take into account there also is the instrumental versus Pavlovian nature of the choices. And again, this controllability factor that I referred to.
Perhaps we have time for one more question. Have you ever investigated the specific role of dopamine modulation in the tail of coded nucleus? There are some emergent evidence in rodents suggesting that dopamine modulation in the tail of striatum, the corresponding structure in rodents, might have a different role there in respect to the most dorsal regions of the striatum. In particular, dopamine modulation there seems to convey more specifically novelty of stimulation rather than reward effort assessment.
Yeah, I know of that work. It's interesting. We don't see that that region commonly light up in our studies. I have to put it that way. We do look at surprise effects or not, but I've never truly manipulated full novelty. So of course, surprise is somewhat different from novelty effects. So perhaps in studies that include that kind of manipulation, such an effect would be revealed, but we haven't addressed that in our studies. Yeah.
Thank you.
And then we have one more last question. Could you speculate a little more on how your final idea about dopamine release being controlled from the frontal cortex could change the food reinforcement field, if it does?
Food reinforcement field, that's a large field. Let me think about how to begin answering that question. So the real the hypothesis or the speculation, I would say it's really rather speculative. It follows from, it's really in line or concurs with a growing body of evidence showing that what these dopamine neurons signal is much more complex than the classic model-free reward prediction error. So of course, as you know, a very pervasive, and as I mentioned, a very pervasive hypothesis is that what dopamine does is modulate model-free temporal difference reinforcement learning, this kind of habit-based stamping in a model-free, a sort of non-cognitive way, right? In a way that is directed at maximizing reward, but doesn't require a model of the structure of the environment in order to get to that goal in novel environment or environment that have changed. More recently, there's accumulating evidence that dopamine neurons and also dopamine release measured in the striatum doesn't simply reflect that kind of maybe dumb reward prediction error, but is reflecting rather more complicated and all kinds of cortical inputs. So the way that this kind of model, and I think more generally the field, can change how we think about dopamine also in the context of food reinforcement, is that it isn't simply, doing reinforcement in the habit sense, but it can be rather clever thing. The intelligence of the dopamine system doesn't originate from this ancient midbrain, but it's rather inherited from all its cortical inputs and subcortical inputs, I should say, and add. And so we're going to have to take into account that higher level processing complexity in order to account for what dopamine does also in food reinforcement.
All right, thank you. Yes, thanks again for your talk and taking your time for us. We have now lunch break until 1:45, and then we continue with our workshop on climate change in neuroscience with Kate Jeffrey and Tom Burling. So it's Margaret.